All right. Last but certainly not least, we have Isabelle Danforth. She received a BS in marine science from the University of California, San Diego, and is currently a second-year graduate student at VIMS. Isabelle is broadly interested in viral evolution, host pathogen coevolution, conservation biology, and molecular epidemiology. Today, she's going to talk about the impact of an IHNV DNA. It's a lot of letters in a row. IHNV DNA vaccine on transmission and virulence evolution in rainbow trout, Oncorhynchus micus. So, Isabelle, it's all yours.
Awesome. Well, thank you for that introduction, Matt. So Melina showed you earlier how it seems as though virulence in IHNV is kind of increasing through time, but rainbow trout are a farmed species where there's a lot of farm-related factors like vaccination that can also influence the kind of evolutionary trajectory of virulence. So I'm going to pivot to talking about how an IHNV DNA vaccine might be influencing transmission and virulence evolution here. Okay. There we go. As a quick refresher, this is IHNV is a negative sense RNA virus in the family Rhabdoviridae. It causes an acute infection in many salmonid species where mortality in juveniles and fry can actually exceed 90% in some cases. And it's transmitted through the water where it causes really rapid outbreaks in high density aquaculture environments. Fortunately, we do have a commercially available DNA vaccine, and very generally, it's kind of synthesized by inserting the G gene into a plasmid vector and then injecting the plasmid into the fish, generally through intramuscular injection, which leads to the expression of the IHNV glycoprotein, which is the most immunogenic of the proteins in IHNV. And we know that the vaccine is really effective at reducing mortality. So that's what I'm showing you here, where you can see that the mortality in the vaccinated group is extremely low, whereas in the unvaccinated treatment groups, there are much higher mortalities. But we also know that the vaccine does not prevent shedding or the release of viral particles into the environment, which is what I'm showing you in the other figure, where you can see that the number of fish shedding that are vaccinated and the number of fish shedding that are unvaccinated is pretty similar up until 3 days post-infection. And these types of vaccines are often referred to in the disease ecology literature as imperfect vaccines or sometimes leaky vaccines. And we can actually turn to evolutionary theory to predict how such imperfect vaccines might influence virulence evolution. So the purple line here denotes the relationship between virulence and fitness that we would expect in this unvaccinated population, where optimal pathogen fitness is achieved at an intermediate level of virulence. But in the case where we have imperfect vaccines being used, which is denoted by this green line, we expect optimal pathogen fitness to require a high level of virulence. And so theory predicts that imperfect vaccines select for more virulent pathogen strains by relaxing the cost of virulence, which we generally assume to be mortality. And we in fact know that this kind of evolution has occurred in chicken agriculture, where there's good evidence that imperfect vaccines have selected for more virulent MDV or Marek's disease virus strains, but we don't know if this is going to happen in trout aquaculture as well. And so for my first chapter, I focused on characterizing the relationship between IHNV fitness and virulence in vaccinated rainbow trout. And I asked the question, will hypervirulent IHNV isolates be more fit than less virulent isolates in vaccinated fish, with the assumption that if they are, then they will be selected for. And I hypothesized that in accordance with the imperfect vaccine hypothesis, we would expect that high virulence isolates will experience a fitness benefit in the context of vaccination. And for the purposes of this project, we defined fitness as the product of transmission rate and transmission duration, where in this system, transmission rate would largely be governed by the quantity of virus being shed and also the probability of shedding or probability of being infected, sorry. And then transmission duration would largely be impacted by mortality. So we conducted 2 experiments to quantify each of these metrics, and both started by vaccinating fish at 1 gram in size with 50 nanograms of the IHNV DNA vaccine or a phosphate buffered saline as an unvaccinated control. And then we waited 30 days at 15 degrees Celsius to allow for the development of an adaptive immune response before challenging groups of 20 fish to one of 5 IHNV isolates or a negative control via bath immersion. These are the 5 IHNV isolates that we selected, and they will look rather familiar to you from earlier talks. We used some of Melina's data to pick these isolates because we wanted to capture the range of possible virulence, and so her data was really useful in deciding that. The first experiment we ran was a virulence assay where we exposed triplicate groups of 20 fish to one of the 5 IHV isolates and then tracked mortality over the course of 30 days. And in the second experiment, we conducted shedding assays where we exposed the fish in the same way, but then transferred them to individual tanks and took water samples on day 3 post-exposure to quantify the amount of viral RNA in the water at that time point. And we used the digital qPCR assay that Hannah mentioned earlier. The first thing I want to show you is some of our mortality data. So here on the x-axis are the 5 IHV isolates, and the y-axis is the predicted probability of mortality as determined by logistic regression, with these individual points denoting the actual data, the proportion of fish that we actually saw die, and then this is the model output with 95% confidence intervals. And if we start by looking at the unvaccinated fish, the purple ones, we can see that we have captured the range of virulence that we were looking for. And so here we see that SV76 caused the least amount of mortality in the unvaccinated fish, but 22090 caused the most. If we overlay that with the vaccinated fish, we see that the probability of mortality is reduced to the same level by vaccination, regardless of isolate virulence in the unvaccinated fish. And so what this could potentially be showing us is that the fitness cost of virulence, which, as you'll remember, is generally assumed to be mortality, has been kind of reduced in this case. But to kind of understand how this would impact pathogen fitness, we need to look at how vaccination also impacts transmission and shedding intensity. And that's what we did with our second experiment. So here I'm showing the shedding intensity on the, I guess it would be the left-hand side of the screen, in the vaccinated, unvaccinated fish, and we saw no effect of virus here. So the intensity of shedding was the same across all viral isolates, but we do see that at peak infection, vaccinated fish shed less virus than unvaccinated fish. If we look at the probability of shedding, in my other figure, we see that there is a little bit more of an interesting story here. So I want to kind of draw your attention to the low virulence isolate SV76, where you can see that the unvaccinated fish are significantly more likely to shed than the vaccinated fish. But that's not the case when we look at the high virulence isolate, where we see that here there's no difference in the probability of shedding. So we kind of haven't figured out what all of this means in terms of the relationship between virulence and fitness, which is unfortunate because that is the first chapter of my dissertation. But I think at some point in the near future, we will get there. At the very least, I think what this suggests is that vaccination could potentially be favoring high virulence isolates, and definitely that we want to look at ways in which we can actually reduce this transmission from vaccinated fish. So I had the good fortune of working with a NSF RU student over the summer. She is from the Universidad de Ana G. Menendez in Puerto Rico, and she was really interested in, can vaccine shedding protection efficacy be improved by increasing vaccine dose? And so she hypothesized that increasing vaccine dose will reduce viral shedding, and that low virulence isolates will experience a greater reduction in shedding intensity with increasing vaccine dose than the high virulence isolates will. So there'll be some kind of virus or potentially virulence effect here. And in this experiment, we vaccinated fish with several doses of the vaccine. So we started with 10, 50, 100, 250, and 1,000 nanograms of the same DNA vaccine. We also, of course, had an unvaccinated control group. And then we challenged the fish to the low virulence isolate SV76, higher virulence isolate 22090, or a MEM10 negative control 30 days post-vaccination. And so we exposed them for an hour and then flushed the tanks for another hour and then transferred them to individual tanks where we took water samples on day 3 and then again quantified those with the digital qPCR assay. And I also want to note that in the case of SV76, we actually only had 3 vaccine doses that were used. So we had PBS, 50, and 250. And in the case of 22090, we had all 6 doses that were used. So here I'm showing you the shedding intensity data for fish challenged with our high virulence isolate 22090. And here I have vaccine dose in order of increasing dose on the x-axis with shedding intensity on the y. And what we found is that there's actually no difference in shedding intensity across all doses. So unvaccinated fish are shedding just as much as those that are vaccinated at 1,000 nanograms of the DNA vaccine. Similarly, we saw no difference in the probability of shedding across all vaccine doses. So unvaccinated fish are just as likely to shed as those vaccinated with our highest vaccine dose. And then if we compare that to the data for the low virulence isolate, SV76, we saw that the unvaccinated fish are shedding more than the vaccinated fish, but within the vaccine treatments, there's no difference. So vaccinating at 50 nanograms is not better in terms of reducing shedding intensity than vaccinating at 250 nanograms. And again, we saw no difference in the probability of shedding across vaccine doses for SV76. And we're also interested in kind of looking at if there's a virus effect here, if these responses would differ by virus. So we trimmed the data set to include those 3 shared vaccine doses and tried to see if we could find an interaction between vaccine dose and virus. There is none. There was no interaction between vaccine and virus, but I think just looking at these plots, you can see that there is maybe some indication that there are different responses between the viruses, though we would definitely need more experiments to kind of get at that. And also, it's not clear whether or not that would be associated with necessarily virulence, or if it could just be, general antigenic diversity between these 2 viruses, which would also require more experiments. So to conclude, I think that we confirm some of the previous results that the IHNV DNA vaccine reduces mortality and also shedding intensity, but it does not eliminate transmission potential. We also found that vaccination does not impact shedding probability for high virulence isolates, suggesting a potential fitness benefit to virulence in these vaccinated hosts. But like I said, I think we don't really know what this means yet in terms of IHNV virulence evolution and what that will look like in the future. I have a couple of experiments in the works at the moment that I think will get at that more directly, and I'm happy to talk about those later. But at the moment, I think that is still kind of an unclear area for us. And then lastly, that Angelica's work found that increasing vaccine dosage does not reduce shedding intensity or probability, and that this relationship does not seem to depend on the virulence of the virus. And so some future work with that would involve seeing if adjuvants could potentially help reduce transmission, and then potentially also pairing this shedding data with virulence assays to make sure that we're getting the same mortality that we would expect to see for a given vaccine dose, and that kind of matches with our shedding data. And with that, I want to thank the Wargo Lab, as well as my committee members and VIMS for funding all of this, and the USDA National Center for Cool and Cold Water Aquaculture for providing the fish that were used in the study, as well as the NSFRU program for allowing me to work with Angelica this summer. So I'll take any questions.
All right, excellent. Nice talk. Very good.
All right, can you still see that, or?
Yep, we're still rolling. If anybody has any questions for Isabelle, same scenario, go ahead, raise your hand, or Mikolash.
Yeah, sorry. Yeah, excellent work. I'm really enjoying the presentations, and it is a tough job to do. My question is more from the point of view of the virus biology. You have a rhabdovirus here, which is a rather small virus, which doesn't have too much of space to develop a lot of tools to evade or let's say evade the host. And you are giving the example from Marek's disease, which is a herpes virus, which is a huge virus, which has a lot of tools and a lot of space to develop stuff. Will you, how are you predicting? Will you get to a real mechanism? And is there any mechanism in which the virus is overcoming the vaccination and somehow developing higher virulence in your case?
That's such a great question, because I've wondered the exact same thing. Definitely, I'm not sure how fair of a comparison it is when we look at, really the only examples we have of imperfect vaccines selecting for high virulence are a parasite, so malaria, rodent malaria, and then also Marek's disease virus. So we really don't know what this would look like for a small RNA virus. So yeah, I'm not really sure. I've thought about that a lot, with limited genome size, the fact that a lot of times most of these genes are serving multiple functions. Yeah, I'm not sure how we can predict that, but I'm excited to see if we can find something interesting.
I encourage you to keep on working because it's a really cool work.
Thank you.
Yeah, and just I think it's amazing, like even though it is a small genome, there's a lot of variation in virulence as you've seen. So even with only 11 kb, it's able to create a lot of phenotypic variation in shedding profiles and virulence profiles. Whether vaccination is going to select on that is a big question, but certainly the variation is there. So yeah, thanks for that.
Isabel, I was curious, you talked about, whether or not vaccine is kind of selecting for more virulent strains, or whether you're even seeing like enhanced virulence. How would you go about testing that? That's actually, I mean, it's a really great question, but I'm curious how you even get to the bottom of something like that. What would you propose to, you have something in mind? Or if you don't want to share that, save that for another presentation, that's fine too.
Well, I hope to eventually get to present on it, since that is in fact the point of my dissertation. But I think one of the projects that I'm most excited about, and one of the most kind of, I think, informative experimental designs in general is experimental evolution. So actually kind of trying to simulate viral evolution in a lab setting, which is kind of a unique, feature for virus host systems, that we can actually watch evolution happen in real time in the tanks themselves. So we spent a lot of time trying to get a serial passage protocol to work. And I think that we're getting there. And so with that, I think the idea would be to try to get the virus to circulate well, not necessarily circulate, but go through multiple periods of transmission in hosts of different vaccine states. And there's just so many options that you could do if you can get something like that to work. And you can actually directly see, when you compare the ancestral viruses to the passage viruses, you can see exactly what happened and how. So
Yeah, with a short genome like
Mhm.
that, you can do some, more resolute screening than with larger genomes, or like with bacterial species or some other things. So
Yeah.
very cool. Looking forward to seeing how this project progresses.
Yeah.
We're coming up on 1 o'clock, so we filled the time. Dr. Wargo and his group, thank you very much. A big round of applause for everybody, and good to see everyone. And we'll be back next week, and hopefully Nick will be back at the helm, so things will go a lot smoother. So thank you, everybody.
It went great, Matt. Thanks, everybody.
Thanks, guys. We'll see y'all soon.
Bye.
Thanks, Matt. Bye.